for the RGR Academy. Students, you can listen to my voice. Students, I think so. Can you listen to my voice? Good morning to everyone. Say good morning in the chat window so that I'll understand you are there in our session. Can you say good morning? Hi, how are you? Good evening to the people who are in some other country. Good afternoon to some other friends who are there in some other country. Good morning to Indian students. Please let me know. Hi, students. Any doubt? Can you start the numerical biology session? You all said, I think so. You all arranged your mic, video, audio, everything, right? Video, audio. Can I go to the session? Hi, students. Good morning. Say good morning. Can you start typing in the chat window? Hi. Can you hear me? Can you hear me? Hi, good morning, students. How many are there, sir? Can you start, sir? Yeah. So now, from RGR Academy, good morning to everyone. Today we are going to discuss the mathematical biology session for the NEET. Okay. Here, let me discuss what type of issues we are going to come across various chapters of grade 11 and 12. Right. Let me start with the grade 12. In the grade 12, we know very well principle of inheritance. So in the case of principle of inheritance chapter, also we can say this chapter as genetics also. So we all know very well about principle of inheritance or genetics chapter. In this, in the NCERT textbook, in the CBSE textbook, we already given monohybrid cross. We already had given monohybrid cross. They already had given monohybrid cross. So what I am going to say for you in this place, okay, in the monohybrid cross, you do any type of cross, not a problem. The parental phenotype should be equal to parental genotype, should be equal to F1 phenotype, should be equal to F1 genotype, should be equal to F2 phenotype, should be equal to F2 genotype. F2 genotype. So understand, parental phenotype should be equal to parental genotype, should be equal to F1 phenotype, should be equal to F2 genotype, should be equal to F2 phenotype, should be equal to F2 genotype. Listen very carefully. Condition 1. Condition 1. In the condition, one parent should be heterozygous. One parent should be heterozygous. So heterozygous. One parent should be heterozygous. And second thing, I don't want the value. I don't want the value. I need only the ratio. I don't want the value. I need only the ratio. Again, back I'm repeating. I don't want the value. I need only the ratio. Right? Okay. So work out. Do any monohybrid cross. You take yellow color, green color, wrinkle seed, anything as you like. Right? And you can ask any questions also in between. Okay, one more time I'll repeat. Do any monohybrid cross. Condition 1, the parent should be heterozygous. Condition 2, I don't want any value, I need only the ratio. Ratio is different, value is different. The ratio for all these things should be same. That means parental phenotype ratio should be equal to parental genotype ratio should be equal to F1 phenotype ratio should be equal to F1 genotype should be equal to F2 phenotype ratio should be equal to F2 genotype. I'll be giving five minutes time for you. Those who are having doubts, please let me know. Those who are having doubts, please let me know. And I'll be solving this question. Can I start solving? Start solving very fast. If you can't do it, I'll be solving it. So please see the question. If you're having any doubt, you can ask me. There are some students who are sitting in front of me also. This is a hybrid session. Some students are sitting in front of me. I am moving around across all the benches. Okay, some students are sitting offline. Some students are there in the online. I'm covering both in this hybrid class. If you're having any doubt, please ask me. I'm there with you to help you. Any students are having any doubt? You know the answer, how to do it? You don't know how to do it? Okay, not a problem. Any students are having any doubt? I given time, sufficient time for you to do it. Nobody done. So now I'm going to do the information for you. Okay, now I'm going to solve this. The thing is, we all know very well, in monohybrid class, if you are taking the heterozygous, hetero, zygous tall we are taking, and then we are crossing with dwarf recessive. If you are crossing dwarf recessive, okay, we can make it very simple. Okay, here dwarf you take homozygous, dwarf you take homozygous, tall heterozygous you take. So capital T, small t, and then you cross with the dwarf small t, small t. So in this case, the phenotype, if you're talking about the phenotype, you know very well, one is tall, another is dwarf. The ratio will be 1:1. If you're talking about the genotype, capital T, small t, and then small t, that is 1:1. So this is the parental phenotype. We know very well the gamete segregation will happen. The gamete I am writing with the circle so that you will understand very nicely. Capital T I am giving circle, there is a gamete. Small t I am giving a circle, there is a gamete, and other small t I am giving in a circle. So this can make two type of gamete, this can make one type of gamete. So what I am doing, I am crossing this capital T and small t, and small t, and again back the small t I am crossing. So capital T, small t you will be getting, and again back small t you will be getting. So what you are doing, you are crossing both the hybrid again back. Before doing the hybrid, let me have the calculation. Here the phenotype will be tall dwarf 1:1 and genotype also 1:1. Again back, this will make two type of gamete. You will be getting two type of gamete. So again back, here also you will be getting the gamete. So if I'm crossing, so you'll be getting capital T, small t, and small t, small t. So here again back, here also I'm getting the tall dwarf and genotype. That means the answer I am bringing finally parental phenotype is equal to parental genotype is equal to F1 phenotype is equal to F1 genotype is equal to F2 phenotype is equal to F2 genotype, which is nothing but 1 is to 1. This is the answer. This is the answer, right? So in the CBSE NEET exam, like this, why we are saying why? Because they are not having any authority. The NEET board is not having any authority to ask question outside the CBSE textbook. But they will confuse the existing CBSE information to the maximum level so that you should not say the answer, so that you will be filtered and you will be not coming for any competition. Okay, so that is the reason why they will ask you, you do any monohybrid cross or dihybrid cross, whatever it may be, but the ratio should be same in all the generation. Parental phenotype should be equal to parental genotype, should be equal to F1 phenotype, should be equal to F1 genotype, should be equal to F2 phenotype, should be equal to F2 genotype, should be equal to 1 is to 1. This is the answer, correct answer. Like this, you can go with other characters also. One will be heterozygous, one will be a recessive type. Okay, and one more question we'll do. I'll be giving some few minutes time to write everything. Okay, if you're finishing it off, I'll go to the next session. Whatever I written in the board, please copy in your note so that I'll erase it and then I'll go to the next important question in the genetics. Then after that, we'll go to plant respiration. Like this, we'll discuss all the different chapters, numericals. Any students, if you're having any doubt, please put it across in the chat so that I'm ready to help you out. Have you completed this writing? Make it fast because we are not having time. Have you completed? Yeah. Have you written this ratio, 1 is to 1 ratio? Students who are in the offline, have you written this 1 is to 1 ratio? This value written? I think so, you're not written. Equal to 1 is to 1, you're not written. Not a problem. Please complete it. I think so, you all written this message. Can I erase the board? Okay, done. So now we are going to the next important topic called dihybrid cross. All of you listen very, very carefully. We know very well dihybrid cross is nothing but a cross made between two organisms varying in two different character. We all know very well about this. What I'm going to do is how I got the ratio. The question is the same question. Parental phenotype should be equal to parental genotype, should be equal to F1 phenotype, should be equal to F1 genotype, should be equal to F2 phenotype, should be equal to F2 genotype, should be equal to 1 value, right? Okay, this is the question they told us to solve, one in monohybrid cross, one in dihybrid cross. So what I am doing for you, for this will form two type of gamete. This will form two type of gamete. You know very well, this is nothing but green round. Green round. This is nothing but green wrinkled. When I am crossing both the parent, you will be getting two different type of gamete with a green round and one type of gamete with a green wrinkle. When I am making or facilitating the fertilization process to happen, what is exactly happening here? You will be getting two different type of progeny. In this two different type of progeny, you will be getting this type of answer. So here we know very well the phenotype of the parent. is nothing but green round and green wrinkle. So this green round and green wrinkle is there in the ratio green round and green wrinkle. Okay, it is in the ratio 1:1. If I take in the genotype, if I take in the genotype value, what I given here, if I am calculating, you will be getting 1:1. So again back, when I'm going for F1 generation calculation, the same result I'm getting. Again back, I'm crossing this two hybrid, whatever I got in the F1 generation. I'm getting two different type of gamete. When I'm crossing, I'll be getting the two different type of progeny in the F2 generation. When I'm crossing the F1 hybrid as a parent. So you'll be getting this type of F2 progeny. So if you're calculating the phenotype and genotype of the parent of the F1 generation or the F2 generation, everywhere we are getting the answer 1:1. This is the answer. So in the NEET exam, they will ask you to bring the same ratio in all the generation. If you are bring the same ratio in all the generation, then your answer will be correct. One in monohybrid cross, one in dihybrid cross. If you are giving the answer as a same ratio, then you will be getting full marks. For getting the same ratio, the way how I worked in the monohybrid cross and dihybrid cross, I already had explained. So the existing monohybrid cross and dihybrid cross, they will confuse us. So be clear enough, be confident enough to solve the issues so that you will get the correct answer. I'll be giving only 2 minutes time because I need to discuss so many other questions of so many other chapters at the numerical biology. This is not the normal monohybrid cross. This is not a normal dihybrid cross. Why? Because we are having a lot of conditions. What condition? We should get the value same for the genotype and phenotype of not only the parent, not only of the F1 generation, including F2 generation as well, not only in monohybrid, even in the dihybrid cross. Anybody having any doubt? Now, after this, we are going for recombination calculation. Recombination calculation, not only meiosis, not only linked allele, not only unlinked allele, not only meiosis process, not only recombination, not only new combination, not only parental combination. We are going to bring all the parameters in one lane. My dear students, anybody having any doubt? I think so. No one is having the doubt. Nobody typed in the chat window. And offline students, have you completed this, both these things? So please copy from others, those who came late. We are not having time. So now, let me go to the next important topic called, see, listen, we need to find out directly proportional, indirectly proportional. We all know very well what is directly proportional. Okay, indirectly proportional. They'll give so many values, so many values. Find the directly proportional, indirectly proportional in the following. This is the question. Okay, now I am writing linked allele, unlinked allele, crossing over, recombination, Prophase 1. Parental combination. New combination. Hybrid. Variant. Hybrid. Variant. Prophase 1. Then has meta formation. So I'm keeping this as A, I'm keeping this as B, I'm keeping this as C, I'm keeping this as D, I'm keeping this as E, okay, I'm keeping this as F, I'm keeping this as G, I'm keeping this as H, I'm keeping this as I, I'm keeping this as L. Now, what you should supposed to do among all these alphabets, which is directly proportional and which is indirectly proportional, that you need to find out. That you need to find out. Which is directly proportional, which is indirectly proportional. Can you start working very fast? Which is directly proportional, which is indirectly proportional. Which is directly proportional, which is indirectly proportional. It's very easy. Okay. If you are giving confusion to the confusion, confusion is not a confusion. Because confusion got confused. So confusion will not remain in a confused state for a longer time. So don't have any tension. Be happy. Just understand the concept, solve it. If you're having any doubt, if you're asking me to repeat the question, I'll repeat the question again back. Actually, in the NEET, these are all the filtration zone. Why? Because it makes you to think much more. It makes you to think much more because these are all the filtration zone. This will make you to think much more. So what will happen? You will consume the time, 5 minutes, 6 minutes time. So what will happen at the time? You will miss many questions. So directly, what I am removing 5, 6 questions, okay, from you, from not attending. So you will not attend. Why? Because you will consume a lot of time in this. Why? Because you don't know the shortcut. You don't know the shortcut. Okay, everybody are working very hard for the long route. So that 5 to 6, 7 minutes you will waste. So that what you will miss 7 questions, 7 to 20 questions you will miss it. Then what will happen? Finally, you will not get the result. That is the logic of making this question. But you should do within 1 minute. Offline students, those who don't know, please let me know. Again, back I'll repeat. Online students, if you don't know, please ask me to repeat so that what I'll explain the concept to you. This is basically will help you to solve any questions of the recombination mapping system. This will make you to solve the questions of meiosis, mitosis. You should get used to it. If you're keeping on solving, then only what you can get the very good score, even if they're confusing to the any level. Concept clarity, if it is there, then we can get the results very fast. I think so. Have you finished it? Have you finished it? Done? You don't know? Or no? Very good. You don't know. You should not take this much time. 180 minutes. In the NEET, only 180 minutes will be given for solving 180 questions. You should keep in this again back 20 minutes break for checking the answer key, for checking the answers, for bubbling the registration number, roll number. So nobody, okay, I can't give much more time. Sorry, your time is over. Now I'm going for discussing about the answers. All of you keep your mind calm enough and listen very, very carefully. The simple logic is A will not decide B, will not decide C, will not decide D, will not decide E, will decide A. A is linked allele and F will decide the parental combination. Okay. And new combination no, hybrid no, variant no, chiasmata no. So A and F are directly proportional with each other. Whereas B, C, D, E, then G, H, I, L. All these things are directly proportional. These two things are indirectly proportional. These two things are indirectly proportional. So unlinked alleles, if it is there much more, definitely crossing over will happen. If the crossing over happens, recombination happens. Recombination happens in prophase one, or the meiosis one, or the meiosis. So definitely there will be no chance for parental combination. Only new combination will be there. New combination and recombination both are same. Both are same. Hybrid and variant both are same. Hybrid and variant both are same. Okay, chiasmata formation happens during recombination. Happens during recombination means if chiasmata happens, then only recombination happens. Right? That means before recombination, chiasmata formation will happen. So that is why the answer is, all of you should understand, okay, the A and F is exactly, the A and F is exactly equal to directly proportional. A and F is directly proportional. Yes or no? And B, C, D, rest everything are directly proportional. Anybody having any doubt? I given suitable sufficient time for you, but no one. Some students written, some students not written. Some students took a lot of time. Anyone having the doubt? Anyone having the doubt? No one is having the doubt. So I already given the solution to you. Now I am going for discussing about the next. In genetics, we discussed three important questions. Three important questions we discussed. Okay, along with the cell division. And now we are going to the next important topic. Okay, if you are seeing this question, in the case of let you work in sexual reproduction in flowering plants, can you see this sexual reproduction in flowering plants? A pollen grain as two male gametes. See here, how many male gametes are present in 150 pollen grains? How many pollen grains are there? How many male gametes are there in 150 pollen grain? So we all know very well, in one pollen grain, we are having one male gamete. Okay, in one pollen grain, we are having one male gamete or two male gamete. Like this, we need to think. We all know very well, inside the pollen grain, we are having one vegetative cell and one generative cell. Inside the pollen grain, we are having one generative cell, one vegetative cell. If one generative cell divides, you will be getting two male gametes. Inside the pollen grain, we are having vegetative cell plus generative cell. Vegetative cell nourishes the generative cell. We all know very well. One generative cell will divide and make how many? Two male gamete. So you all need to understand that one generative cell is not undergoing meiosis. That one generative cell is not undergoing meiosis. That one generative cell is undergoing haploid mitosis. Is totally different from diploid mitosis. When one generative cell of the pollen grain or microspore, if it is getting divided, it makes two haploid male gamete. Why, sir, you are saying haploid mitosis? Why? Because the mother cell, which is participating in the pollen grain division, the mother cell, which is nothing but the generative cell, of the pollen grain, when it is undergoing mitosis process, you will be getting two male gamete. You will be getting two male gamete from one generative cell. Generative cell is also haploid. Male gamete is also haploid. That is the reason why we are saying haploid mitosis. We are saying haploid mitosis totally different from diploid mitosis. Totally different from diploid mitosis. So that is the reason why the pollen grain contains how many male gamete? If we are having 150 pollen grain, in 150 pollen grain, how many male gametes? 150 into 300. Are you clear? Any doubt is there? Any doubt? Children's? Any one of you are having doubt in this place? Can I go to the next question? An ovule has one egg and two polar nuclei. How many polar nuclei are present in 50 ovule? We all know very well, in the one ovule, nuclei, polar nuclei means plural. They told, listen very carefully, in one ovule, they are saying nuclei means they are indicating not secondary polar nucleus. They are indicating only primary, only indicating primary. Primary polar nucleus are two in number. In one ovule. That means 50 ovule will be having how many? 100. 50 ovule will be having how many? 100. One ovule is having two primary polar nucleus. That means 50 ovule will be having how many polar nucleus? 100 polar nucleus. Are you clear up to this point? Now, how many male gametes are required to fertilize 80 ovules? 80 ovules. Here, we need to understand, in one ovule, one egg is present. Why we are saying two male gamete? Why we are saying two male gamete? One male gamete is enough to fuse one egg. Why you are saying two male gamete? Why? Because we all should understand the triple fusion process. We know very well in the triple fusion what is exactly happening. One primary polar nucleus fuses with another primary polar nucleus, so that you are getting secondary polar nucleus. If one male gamete fuses the egg, you are getting zygote. If another male gamete fuses the secondary polar nucleus, then only you will be getting endosperm. Are you getting my point? How I got the endosperm? I got the endosperm because of the fusion of secondary polar nucleus and one male gamete. How we got the zygote? Because of the fusion of one egg along with the one male gamete. How we got the secondary polar nucleus? Because of the fusion of two primary polar nucleus. By keeping these concepts, these means plural. By keeping these many number of concepts in the mind, I am saying in one ovule, two male gamete. For 80 ovule, 80 into 2, 160. I need 160 male gamete. For solving this question, you need to know double fertilization, you need to know triple fusion in the ovule, right? So now we solve three questions. Now let me go for let you go for the board work. Right? Can you go to for the board work? Right. The simple logic is, listen very carefully. Okay? For making 168 zygote, for making 168 zygote, I required, for making 168 zygote, I required how many synergid, how many primary polar nuclei I required, how many secondary polar nucleus I required, how many egg I required, how many generative cell, how many generative cell I required. I required how many generative cell, I required how many synergids, I required how many primary polar nucleus, how many secondary polar nucleus, how many egg, I required how many antipodals, I required how many antipodals, right? So I required how many male gamete, I required how many male gamete, right? How many synergids? Say, okay, secondary polar nucleus. Or second, actually, we will be getting only one secondary polar nucleus. Why? Because it is singular. Okay, in none of the ovule, we are having many secondary polar nucleus, except multinucleated. Multinucleated means in the sense multi-seeded fruits like lemon, orange, like this, or pomegranate like this. Okay, forget about those exceptionals. In the exceptionals only, we should supposed to use secondary polar nuclei. Whereas in most of the fruits, most of the ovules, we should use one secondary polar nucleus. Right? Start solving. For making 168 zygote, how many synergids, how many primary polar nucleus, how many secondary polar nucleus, how many egg, how many generative cell, how many antibodies, how many male gamete, how many mitosis is required, how many meiosis are required. So you need to calculate how many meiosis, how many mitosis, how many generative cell, how many antibodies, how many male gamete, how many secondary polar nucleus, how many egg, how many primary polar nucleus, how many synergids. This is called mathematical biology, right? In the sexual reproduction in flowering plants. Before this, we discussed genetics and cell division. Genetics and cell division together we discussed in the numerical biology. Now we are discussing sexual reproduction in flowering plants along with cell division. Right? These type of hybrid questions will come in the NEET exam. Children's, if you are having any doubt, please ask me. Those who are having doubts, please ask me. I am there with you to help you. This is my workstation, workplace. So we are having aggressive numerical biology session in our RGR Academy in the upcoming week. Those who are interested can come and join us. Right? For you also. Yes or no? So then only you will practice a lot of numericals. Why? Because 10 questions will come, definitely numericals questions will come out of 90 questions in the NEET. I given sufficient time. You're taking a lot of time to solve. So now the time is over. Why? Because you crossed more than 2 minutes. Sorry. Please listen to me. How I am doing it. It's very simple. Very simple. I'll work out in this place. See, listen. For making one zygote, they won't give any value for you. For making one zygote, I require two synergids. For making one zygote, I require two synergids. I require two primary polar body, two primary, not polar body, polar nucleus. Polar body, the word you should supposed to use only in the oogenesis process. So, two primary polar nuclei, two primary polar nuclei, okay, and then polar one secondary polar nucleus, one secondary polar nucleus, okay, one egg, okay, and then you need to understand mitosis. I required how many in number? 3 mitosis for making one zygote, okay, 3 mitosis are required for making these many number of cells, okay, and I required one meiosis process. Okay, how I am saying this 3 mitosis, one meiosis? Just take your mind to the sexual reproduction in flowering plants chapter. There we discussed for making 8-celled embryo sac. For making 8-celled embryo sac, we require 3 mitosis and one meiosis. They are already given in the NCERT textbook. By keeping that formula, they will bring any type of question, right? So one mitosis, one meiosis, two generative cell. Like this, you should not supposed to say only one generative cell. One generative cell. One generative cell. 3 antipodals. Antipodals are formed, but it is not supporting zygote formation. But it is formed along with this one. In the case of male, we should supposed to know sometime. Okay, here antipodals, I already told this message for you. And then if you're talking about male gamete, how many male gamete I required? How many male gamete I required? I required 2 male gamete. I required 2 male gamete. This information we need to keep in our mind. This information we need to keep in our mind for solving the question. Now, one question I will solve for you. Remaining things we can solve very easily. Okay, all the things. Totally, how many parameters they asked? Totally 12 parameters they asked. So what I will do, I will work out on synergids. I will work out on synergids for making one zygote. For making one zygote, I require two synergids. For making one zygote, I require two synergids. Right? For 168 zygote, how many synergids are required? So 168 into 2. How much? 8 2s are 16. 6 2s are 12 plus 1, 13. So 2 2s are 2 plus 3. Am I correct? 336. 336. Right? 8 2s are 16. 6 2s are 12 plus 1, 13. 1 2s are 2, 336. Am I correct or not? So now, if you're going for, so this is the answer. So like this, you can calculate. Like this, you can calculate for all the information. Yes or no? You can calculate for not only for synergids, not only for zygote, not only for primary polar bodies, not only for secondary polar bodies, not only antipodals, okay, not only egg, okay, not only generative cell, and we can say how many double fertilizations also are there. How many double fertilization are required? Double fertilization. For 168 egg, we require 168 double fertilization. For 168 zygote, how many fertilization? 168 double fertilization. How many triple fusion? 168 triple fusion. Yes or no? So for making 168 zygote, I required 168 double fertilization. 168 double fertilization. Okay. For making how many triple fusion? I required 168 triple fusion. Triple fusion. Triple fusion. So what they will do in the NEET exam, they will confuse you. How many fertilization are required? For making 168 zygote, I required 336 fertilization. 336 fertilization. 336 fertilization. For making 168 zygote, how many fusion I required? 336 plus 168. Okay, means 14 plus 1, 10, 10 plus 1, 504. 504. 504 fusion I required. 504 fusion I required. How many? 504 fusion I required. How many fertilization I required? 336 fertilization for making 168 zygote. Yes or no? Fertilization versus this problem. Triple fusion versus this problem. Like this, so many questions will come. Right? I think so. You understood the question. Can I erase a board? Doubt? Have you written all this value? Have you written all this value? Have you written? Okay, I'll wait for you. Students, those who not written the value, please write it fast. We need to discuss furthermore other questions. Anybody having any doubt? Students who are having doubt, please let me know. Students who are having doubt, please type in the chat window. So nobody asks any question. Okay, now I'm going for the next important question with other chapter. We discuss questions in sexual reproduction in flowering plants. Now let me go for discussing the human reproduction. Can you see this? In the human reproduction question, can you see in the human reproduction question, from 120 primary spermatocyte, number of sperms formed is how much? 480. 480. Okay, 480. What is the reason? What is the reason? We know very well from the one primary spermatocyte. From one primary spermatocyte, listen very carefully. From one primary spermatocyte, we will be getting 4 spermatozoa. 4 spermatozoa. How many you are getting? 4 spermatozoa. Yes or no? How many spermatozoa you are getting? From one primary spermatocyte, you will be getting 4 sperm cells or 4 spermatozoa from 4 spermatid. We know very well primary spermatocyte undergoes meiosis 1. Because of that, you are getting secondary spermatocyte. And secondary spermatocyte, when it is undergoing the meiosis 2 process, you are getting 4 spermatid. And that 4 spermatid is getting matured or getting differentiated or getting evolved to the spermatozoa, right? So, for making 4 sperm cells, I required 1 primary spermatocyte. That means 1 meiosis I required, 1 primary spermatocyte I required for making 4 spermatozoa or 4 sperm cells. This is the concept. By keeping this concept, 120 into 4, 480 is the correct answer. 480 is the correct answer. Now, how many secondary spermatocyte produced from 75 primary spermatocyte? We all know very well exactly from 75 how much you are getting. Exactly, you are getting how many? From 1 you are getting 2. From 1 you are getting 2. 150 is the correct answer, right? What is the correct answer? 150. Why? 1 primary spermatocyte makes how many secondary spermatocyte? 2. 2. Because of meiosis 1. Because of meiosis 1. See, 75 primary spermatocyte makes secondary spermatocyte by meiosis process. 1 primary spermatocyte makes 2 secondary spermatocyte because of meiosis 1 process. That means 75 will make how many? 150, right? Okay, that means primary spermatocyte is a diploid cell. Secondary spermatocyte is a haploid cell. Primary spermatocyte is a diploid cell. Secondary spermatocyte is a haploid cell. Are you clear? Any doubt? Any doubt? Now, so in the human reproduction, number of spermatid formed from 50 spermatocyte. So, 1 secondary spermatocyte will make how many? 1 secondary spermatocyte will make how many sperm cells? 2. 2 secondary spermatocyte will make how many? 4. 50 will make how many? 100. So these are all easy questions. These are all easy questions. But there are some tough questions are there that we'll discuss furthermore in detail. If you're talking about 400 sperms are formed, number of primary spermatocytes required how many? How many spermatocytes are required? 400 divided by 400 divided by 400 divided by 4. How much you will be getting? 100. Is that correct answer? These are all easy questions. These are all easy questions. To this level, easy questions they won't ask. They will always ask at the tough level. What tough level they will ask? Hmm. From one secondary spermatocyte. From one secondary spermatocyte, you will be getting 2 sperm cells. From 2 secondary spermatocyte, you are getting 4 sperm cells. See, I'll write the information for you. You not to worry. So, some of the offline students are asking some doubt. Online students, please listen to me when I'm explaining about spermatogenesis process for getting the clarity. Again, back spermatogonium. We all know very well, which is a sperm mother cell, which is a diploid cell having the complete number of parental chromosome. When it is undergoing mitosis process, you will be getting 2 primary spermatocyte. 2 primary spermatocyte. So 2 primary spermatocyte you are getting. So I am taking now only 1 primary spermatocyte for calculation. Okay. I'll take only 1 primary spermatocyte and then I am taking this 1 primary spermatocyte for getting entry into the meiosis process. Because of the meiosis 1 process, you are getting how many secondary spermatocyte? You are getting 2 secondary spermatocyte. These 2 secondary spermatocyte, when it is undergoing the meiosis 2 process, this is meiosis 1 process. Because of the meiosis 2 process, these 2 secondary spermatocyte will be making 4 spermatid. Will be making 4 spermatid. We know very well the formation of the spermatid. They are not given in the NCERT textbook, but there is a chance for asking in the board exam. Spermiotelosis process is nothing but formation of spermatid. The spermatid is becoming spermatozoa. Okay, spermatozoa formation or sperm formation or mature sperm formation is nothing but spermiogenesis. Spermiogenesis plus spermiotelosis together is called spermatogenesis process. Okay, spermatogenesis process. So again, back I'll repeat. One spermatogonium is forming 2 primary spermatocyte by mitosis process. The second spermatocyte is also diploid. I am taking from the 2, I am taking only 1. That 1 primary spermatocyte, when it's undergoing meiosis 1 process, you are getting 2 secondary spermatocyte. When this 2 secondary spermatocyte is undergoing meiosis 2 process, you will be getting 4 spermatid. This 4 spermatid is getting matured into 4 spermatozoa by a process called spermiogenesis process. Formation of spermatid, spermiotelosis. Formation of spermatozoa, spermiogenesis plus spermiotelosis together is called spermatogenesis. These 3 terms don't get confused with spermiation process. Spermiation is nothing but release of the sperm, mature sperm, after getting nourishment from Sertoli cells. Okay, all the terms are not similar, right? Release of the mature sperm cells from the Sertoli cell is called spermiation process. That concept, you don't bring it here. That will happen after all this event. After all this event. Clear? Anybody or having any doubt? Please let me know. I'll be going to the next important segment. So you asked the question I told. Okay. Now, the simple, the human reproduction, few questions we discussed. Many varieties of questions we discussed in the case of human reproduction. Let me go to the next important chapter, evolution. You know, in the evolution, Hardy-Weinberg law. You know Hardy-Weinberg law, right? Any one of you can say the Hardy-Weinberg law? Those who are in the offline? Those who are in the offline, please let me know. Those who are in the Hardy, those who know the Hardy-Weinberg law, please let me know. Okay. The simple logic is, in the Hardy-Weinberg law, we all know very well the a plus b whole square is equal to a square plus 2ab plus 2ab plus b square. What we studied in the lower grade, the same thing they are using as p plus q whole square is equal to p square plus 2pq plus q square. The p square exactly explains about dominant homozygous. The q square explains about recessive homozygous. 2pq represents dominant heterozygous condition or recessive heterozygous condition. 2pq represents dominant heterozygous condition or recessive heterozygous condition. Q square recessive homozygous, P square dominant homozygous. So by keeping this Hardy-Weinberg equation, if it is not equal to 1, if it is not equal to 1, what are the 5 probabilities we studied? One probability is genetic shift, second probability is genetic drift, third probability is natural selection, fourth probability is very good recombination, fifth probability mutation. Very good. So these are all the 5 different probabilities we already had given in the case of NCERT textbook. Okay, getting my point? So if it is not equal to 1, the reason for deviation in the population that we are going to calculate. This Hardy-Weinberg law exactly speaks about population genetics concept of evolution. You just commonly understand, okay, the p value they already had given 0.1. Always we should mention one value. Yes or no? P plus q is equal to 1. So q equal to 1 minus 0.3 is 0.03. So the value of the q is 0.3. The value of the q is 0.3. What does this q represents? What does q represents? What does q represents? Recessive allele. Recessive allele. A single recessive allele. A single recessive allele is 0.3. Is 0.3. Clear? Any doubt in this question? A single recessive allele is 0.3. Actually, in few cases alone. Actually, single recessive allele will not bring the character. A single recessive allele will not bring the character, but there are some exceptionals are there. Where, sir, can you say the exceptional? A single recessive allele will bring the character without any other recessive allele. Okay, that is called hemizygous. That is called not homozygous, not heterozygous, hemizygous. Can you give the example? Color blindness. Color blindness. Color blindness, in the color blindness concept, a single recessive allele is enough to bring the issue. Is enough to bring the issue. We don't want two recessive allele. A single recessive allele is enough to bring the issue. That condition is called hemizygous, but that we can't implement everywhere. Okay, there is exceptional cases. The exceptional cases. Done. Now, one more question we'll discuss. In this question, they'll give the p value, they'll ask you to find out q value. This is very easy. Always you keep the concept p plus q is equal to 1. So 1 minus 0.4 equal to 0.06. Okay, actually, we need to understand if your p plus q whole square equal to p square plus 2pq plus q square, like this you are saying. If you remove the square everywhere, p plus q square, if you are removing, it will become 2p plus q, right? Then p square will become p, 2pq will become pq, and q square will become only q. If that is the condition, that p plus q is equal to 1, I am keeping always the p plus q. See, always, for example, 2 different variants are there. Let us consider. Dominant is also there, recessive is also there in a population. I am keeping that entire population as 1. For example, let us consider, I am preparing 80% methanol solution. 80% methanol solution, you are calculating along with 100 ml. Okay, 80 ml of pure methanol and 20 ml of distilled water. That is nothing but what? 80% methanol. 20% methanol, 20 ml of methanol plus 80 ml of distilled water. Like this, you are calculating for 100%. Like this, here you are calculating for 1 is equal to 100%. 1 is equal to 100%. If you need to bring in terms of percentage also, you can bring it in terms of percentage. For example, instead of saying 0.6, instead of saying 0.6, you can say 60%. In a population, how much percentage? 60%. 60%. Okay. In this, can you see? Instead of saying 0.3%, you can say 30%. 30% of the overall population. Sometimes they'll ask in percentage. Please express the final result in percentage. Like this, they're saying you should be in a position to mention the percentage calculation as well, right? Now, they'll give p value, they'll ask you to find out q value. Okay, they'll give q value, they'll ask you to find out p value. They'll give the p value, they'll ask you to find out p square, right? So you should be competent enough to do all this Hardy-Weinberg question. These are all the simple questions. Okay, let me go to molecular basis of inheritance. So, in the molecular basis of inheritance, we know very well about the Chargaff rule. Yes or no? Can you say what is Chargaff rule? What is Chargaff rule? What is Chargaff rule? Huh? 8 plus? Okay. So that you know very well. Now, your 20% cytosine is there. 20% cytosine is there. The 20% cytosine is equal to 20% guanosine. And remaining 60% adenosine thymine pair. Adenosine thymine pair. In this 30% adenosine, 30% thymine. That is the Chargaff rule formula. You know now? Adenine, thymine, guanine, cytosine. Guanine, cytosine. Adenine, thymine, guanine, cytosine. Yes or no? Okay, now let me calculate. Let me calculate for you take rotavirus. You take rotavirus. What virus? Rotavirus. Okay. In this, if cytosine is 40%. If cytosine is 40%, calculate other nucleotide. Calculate other nitrogen base. Calculate another nitrogen base. I told in rotavirus 40% cytosine. In rotavirus, 40% cytosine. Calculate other nucleotide. You should give the value for all the nitrogen bases. You should give the value for all the nitrogen bases. One more time I'll repeat. Rotavirus is in cytosine 40%. You should calculate other nitrogen base. The nitrogen base you should give in a figure. You calculated? Say. Cytosine how much? 40. Guanosine 40. Adenosine 10. Thymine 10. So what is your name? Huh? Sound. Nityashree. So Nityashree told one of the nice wrong answer. So this is wrong. One of the nice wrong answer. This is wrong. Why? Why this is wrong? See, Chargaff rule is not applicable for single strand RNA. Chargaff rule is applicable for double strand RNA. Rotavirus is having double stranded RNA. So where we can apply Chargaff rule. That means cytosine 40, what she told is correct. Guanine 40, what she told is correct. And she told adenine 20%, adenine 10% she told correct. Okay. And no thymine, it is only 10% uracil. Why? Because rotavirus is having double stranded RNA. Rotavirus is having double stranded RNA. Or getting important or not. This is the way they will cheat you. This is the way they will cheat you. What they will do, you know? Okay. In the NEET exam, what question will come, sir? What question will come? Listen very carefully. Cytosine, guanine, adenine, they will not bring uracil concept. They will not bring uracil concept. Thymine. 1, 2, 3, 4. 4 is none of the above. 4 is none of the above. None of the above. Okay. 40, 20, 40, 20. 20, 20, 40, 40. Okay, 40, 40, 20, 20. Answer is what? Answer is what? None of the above. Answer is what question will come, sir? This is the question will come. None of the above. Why? Because in rotavirus there is no thymine concept. There is no double stranded DNA. There is no double stranded DNA, right? So none of the, okay, what you will do? As with the Chargaff rule, in the case of rotavirus, what will happen? Like this, you will calculate. Sir, this is the correct answer. What you will do? You will choose. What you will choose? What you will choose? For example, let us consider. I am giving the answer like this. Okay, all the good students will choose option 3 as the correct answer. Option 3 as a correct answer. Yes or no? You will be trapped. You will be trapped. Right? So, here formula alone will not help you. Biology concept also will help. That means the mathematician, pure mathematician with the formula, they can't play the games here. The pure mathematician with the formulas, they can't play the games. Don't think, sir, we'll substitute formula, sir, we'll get the result, sir. No. You should have the concept, rotavirus is having a double-stranded RNA. Right. Now, anybody having any doubt in this place? I've written this message. So none of the above is the correct answer. Okay, now we are going for the next important other questions in the case of Chargaff rule. Okay, you saw this is the Chargaff rule based questions. Yes or no? Anybody having any doubt in this place? Which one? Yeah. Nature of the DNA or RNA. For see, they can't bring all the viruses, all the things. What all the things are there in the case of NCERT textbook? Those viruses alone they will ask. They are not allowed to use the NEET question paper setters or not having rights to ask any question outside the NCERT textbook. Only see, I am having one. Okay, I am having only rights to ask only inside the NCERT textbook. I can confuse you to the maximum level. Okay, here you asked about, for example, let us consider if you're talking about the HIV. You will be thinking that it is having double-strand RNA. No, it is having 2 single-strand RNA. HIV is having 2 single-strand RNA, not double-strand RNA. Like this, a lot of logics and logical things are there. Now we are going to calculate the length of the DNA and number of base pair. So the question, the distance between the 2 consecutive base pair in the DNA is 3.4 angstrom or 3.4 nanometer. Calculate the total length of the DNA given the number of base pair or vice versa. So a DNA molecule having 10,000 base pair, let us consider. What is the length in the micrometer? So here the length per base pair is 0.34 nanometer, is nothing but 0.34 into 10 to the power of minus 9 meter. The total length of the base pair equal to length per base pair, 10,000 into 0.34 into 10 minus 10 to the power of 9 meter. This is the value you will be getting. So we need to convert finally the meter into micrometer. Finally, meter into, we need to convert into micrometer. So this is the value. This is the value. So you understood this question? If you are not understanding this question, see, listen again back, I'll say to you, length per base pair is different and number of base pair is different. Number of base pair, they already had given how many? 10,000 they given in the question. Number of base pair, how much they given? 10,000. Number of base pair in the book, what they given in your book? Number of base pair in one term, 10 base pair. 10 base pair they given. Sometimes they will not give the base pair per turn. Okay, they will say calculate by keeping 0.34 nanometer between the 2 base pair and number of base pair 10 base pair per turn. Then you need to calculate. Okay, by keeping number of base pair per turn and by keeping the distance between the 2 base pair. They will not give the value 0.34 nanometer. They won't give, or angstrom they won't give. They won't give number of base pair per turn. By considering number of base pair per turn and by keeping the distance calculation between the 2 base pair, calculate the length of the DNA, like this they will say. Okay, we can't go and fight with them. Why? Because in the NCERT textbook, they give only 10 base pair per turn. That is only for human being. That is bDNA. That is bDNA. If we're talking about zDNA, zDNA is having 12 base pair per turn. But that is not in the NCERT textbook. What they will say, in the NCERT textbook, only 10 base pair they given. No, some DNA will be having 11 base pair, they won't listen to you. Some DNA will be having 12 base pair, they won't listen to you. Why? Because in NCERT textbook, they given one turn of the DNA is having 10 base pair per turn. But they're not told this DNA is bDNA. This DNA is human DNA. Like this. So that I am there with you to help you. Again, back one more time, I'll repeat. The plant needs 6 NADPH in Krebs cycle. How many pyruvate oxidation are required? How many glycolysis are required? How many glucose are required? How many acetyl-CoA are required? How many pyruvate are required? Start calculating very fast. Those are any doubts, please ask me. One more time, I'll repeat. If the plant needs 6 NADPH in the Krebs cycle, how many pyruvate oxidation are required? How many glycolysis are required? How many glucose are required? How many acetyl-CoA are required? How many pyruvate are required? Have you started calculating? Hello? If you are giving confusion to the confusion, confusion is not a confusion. Because confusion got confused. So confusion will not remain in a confused state for longer time. Do it. Students who are in the online, please let me know. Why? Because students who are in the offline already completed and they shown the results. Students who are in the online, start posting your calculations and type properly in the chat window so that we are ready to help you out. If you can't solve, I'll solve it for you and I'll say how to do it in a very easy way, in a simplified way. That is why the RGR Academy team is here to solve all the tough NEET biology questions to help the students for getting 360 out of 360. Now time is over. So now I'll be saying the answer for you. Listen very, very carefully. For making simple logic is, okay, here in one Krebs cycle, listen very carefully. In one Krebs cycle, all of you listen very carefully. In one Krebs cycle, for making 3 NADPH, okay, 3 NADPH, I required 1 acetyl-CoA. I required 1 acetyl-CoA. Okay. I required off glucose. I required off glucose. I required off glucose. I required 1 pyruvate. I required 1 pyruvate. Okay. So what we'll be doing, and I required off glycolysis. We should not supposed to say off glycolysis, so that what we are, okay, just we'll write it down and then I'll say the easy way to calculate, right? So There is no off glycolysis in the world. There is no off glucose in the world. So that what I'm doing, if I'm keeping this as, if I'm keeping this as 1, if I'm keeping this as 1, of course, I should suppose to bring pyruvate, acyl-CoA and Krebs cycle as 2, right? So 2 pyruvate, 2 acetyl-CoA, 2 Krebs cycle. 1 glycolysis, 2 pyruvate, 1 glucose, 2 acyl-CoA, 2 Krebs cycle, right? So that means they already had given 6 Na, and how many NADPH will be formed for 2 Krebs cycle? How many NADH will be there? Means 6 NADPH. 6 NADPH. So 6 NADPH. This is the value, right? 6 NADPH. Like this, we should not supposed to say. We should supposed to say only 3 NADPH. So if you need to get 6 NADPH, 3 into 2, 6. Okay, 3 into 2, 6. So 2 into 1, 2 glycolysis. So for achieving this, I am multiplying with 2. So 1 into 2, 2. 2 into 2, 4. 1 into 2, 2. Okay, 1 into 2, 2. 2 into 2, 4. 2 into 2, 4. 2 into 4. So for making 6 NADPH, I require 2 glycolysis, 4 pyruvate, 4 glucose, 4 acetyl-CoA, 4 Krebs cycle. 4 Krebs cycle. This is the answer. This is the answer, right? This is the answer. Right? Right? They in the question what they given? In the question they given to calculate for 6 NADPH. They given 6 NADPH. For 6 NADPH, how many glycolysis? 2 glycolysis. How many pyruvate? 4 pyruvate. How many glucose? 2 glucose. How many acetyl-CoA? 4 acetyl-CoA. How many Krebs cycle? 4 Krebs cycle. Right? Have you completed this? She's writing. I'm waiting for her to complete. Have you completed it? Can I erase the board? Right. So now, listen very carefully. We are going for calculating the ATB. We used to say in IB curriculum, International Baccalaureate curriculum, IGCSE curriculum, and the CBSE advanced curriculum, we used to say energy calculus. Energy calculus. We should go for energy calculus. So, in this energy calculus, let me work out. Listen, one logic is, first of all, you should understand what is oxidative phosphorylation. Oxidative phosphorylation means formation of ATP in mitochondria alone. Formation of mitochondria alone. Okay, with the help of NADH or NADPH, FADH2. Okay, formation of ATP in the oxidative phosphorylation happening because of only these two blocks. That is called oxidative phosphorylation. Substrate level phosphorylation. Substrate level phosphorylation. Substrate level phosphorylation means ATP formed other than mitochondria in the metabolism. The ATP is formed in the metabolism other than mitochondria. Then we are calling this as substrate level phosphorylation. Substrate level phosphorylation. What is meant by photophosphorylation? Photophosphorylation. Like this, you divide the word. If you're having difficulty, need not to worry. The ATP formation because of the photosynthesis or because of the sunlight exposure. That is the reason why we are saying ATP formed with the help of light. ATP formed with the help of light. Okay. So here oxygen is used in oxidative phosphorylation. Oxygen is used in phosphorylation, right? So for doing the energy calculus, we should know all this information. For doing the energy calculus, we should know all this information. So now, I think so. You've written all these 3 points. Okay, first point, then second point, then the third point. Have you completed this? Have you completed this energy calculus 3 concept? Then I'll say one more concept. Then after that, we'll go to the various calculations. Have you completed it? What are? You need the expansion. Actually, they are hydrogen donors. These two are there, no? These two we used to call as hydrogen donors. Hydrogen donors. These hydrogen donors will donate the hydrogen to activate ATP synthase enzyme. To activate ATP synthase enzyme in mitochondria. The ATP synthase enzyme of mitochondria are activated because of the hydrogen given by these people. That is the reason why they are called as hydrogen donors, also called as reducing equivalents. Reducing equivalents, right? Anyone of you are having any doubt? Can I erase the board? Can I go to the next concept? Can you go for solving the problem? Right. So now we're talking about one ATP molecule will give rises to minus 1 mole of ATP. I not told one molecule. One molecule means wrong. 1 mole of ATP give rises to minus 7.3 kilocalories. Kilocalories. Instead of writing like this in the case of NEET exam, when they are giving the question at the time, they will say K dot cal. K dot cal. So how to convert this kilocalories into kilojoules? Just we need to multiply this kilocalories into minus 14.18 or 14 point or minus 4.2, not 14, minus 4.2. So automatically you'll be getting the value, the x value you'll be getting, right? You'll be getting the value. Minus represents what? Exothermic reaction, heat giving reaction. That is why it is called what? Minus reaction or exothermic reaction, also called as exergonic reaction. So exergonic reaction or exothermic reaction, also called as heat leaving reaction. That is why we are representing this with a minus symbol, right? So this will give the kilojoules. This will give the kilojoules value. Okay, kilojoules value. We are converting kilocalories into kilojoules. So for converting kilojoules into kilocalories, what we need to do is we need to multiply or divide. We need to divide. You'll be getting the x value that we need to express in terms of kilocalories. In terms of kilocalories, right? Right? Can I erase the board? Have you completed this, students? You written all these points? Hello? Offline students? Have you completed? Can I erase the board? So now at least we'll solve one problem. Actually, there is the experimentally found value. But there is a value we need to keep it in our mind always. For example, let us consider, we are having some formulas for converting Celsius into Fahrenheit. Okay, Celsius, Fahrenheit, and then Kelvin. Celsius, Fahrenheit, we can convert. Okay, and also you know very well 1000 ml is equal to how many deciliter? 10 deciliter. 100 ml is equal to 1 deciliter. Like this, they kept one defined value by doing the experimental procedure. This is a universal value. Sometimes they will say 4.18, sometimes they will say 4.2, both you accept it. 4.18 or 4.20, both are same. Now let me go to the important calculation. Okay, if one glucose is entering into aerobic glycolysis, how many ATP will be formed? How many ATP will be formed by substrate level phosphorylation? How many ATP will be formed by substrate level phosphorylation? Okay. This is the answer. So here they told substrate level phosphorylation. We should not supposed to calculate, we should not supposed to bring NADH, we should not supposed to bring FADH concept. In glycolysis, there is no FADH2. NADH concept, we should not bring it. Why? Because they told substrate level phosphorylation. So the gross ATP formed are 4. Okay, and how many ATP is used in the glycolysis is 2. So you'll be getting a net. So in the NEET exam, they're asking how many ATP will be formed by substrate level phosphorylation. You should understand if they are using the word gross, there is the word gross, it is 4. If they are saying net, it is 2. Minus 2 is what? Used. Minus 2 is used. For example, I'm working in Canada. Okay, my salary is 5 lakhs. My salary is 9 lakhs. How much tax I'm paying? I'm paying 2.5 lakhs tax. Okay, my take home salary is 6.5. So the 4 is what? Full salary, minus 2 is tax, and 2 lakhs is take home salary. Right? Like this, you need to remember the gross ATP which is formed in your glycolysis process, aerobic glycolysis process, is nothing but what? 4. And how much ATP is used? Minus 2. Okay, 2. Because they told substrate level phosphorylation. Substrate level phosphorylation. Right? Like so many sums are there in the case of plant respiration. Okay, with this, they will trap you not to get 360. Okay, 360, because everybody are learning very nicely the NCERT textbook. I need to bring the confusion. So you'll be trapped. Finished? If they're asking you to calculate, the kilocalories, how you will calculate? Just we need to multiply with 7.3. Just we need to multiply with 7.3. If they're asking you to calculate the x value, they're asking you to calculate kilojoules. Again, the x value you multiply with 4.18. Again, back you multiply with 4.18, you will get the final y value. The y is the correct answer. Y is the correct answer. For example, they will ask you to calculate. They'll give all these conditions. They'll ask you to calculate kilojoules. They'll ask you to calculate kilocalories. So kilocalories means multiply with 7.3. And whatever the value you are getting, that you multiply again back with 4.18 to get the kilojoules. Sometimes they will ask you to explain, sometimes they will ask you to say in only calories. Calories. You know very well 7.3 kilocalories is equal to 7,300 calories. Right or not? Right? Why? Because 1 kilocalories is equal to 1000. So you should know how to convert kilocalories into calories, calories into kilocalories, kilojoules into joules, joules into kilojoules. Right? You know how to calculate this? Okay, first they told how many gross ATP will be formed by substrate level phosphorylation. They told gross. Gross means you need to give 4 value. They're saying net. You should give 2 value. You should give 2. Sir, they not mention gross also. They are not mention net also. Then you need to mention only net. Then you need to mention only net. Sir, they not mention gross also. They not mention net also, sir. Then you go for net. If they ask you to calculate kilocalories, you multiply with 7.3. If they ask you to say kilojoules, you mention with this. Are you clear enough up to this point? So now, if they are giving 50,000 kilojoules are formed in aerobic glycolysis. Aerobic glycolysis, you should calculate how many glucose participated. How many glucose participated? How many glucose participated in aerobic glycolysis? This is the homework for you. Directly no. First you should convert into kilocalories, then you convert into kilojoules. First you convert it, first you get kilocalories, then you go for kilojoules. Okay, and also here also in aerobic glycolysis, first you calculate what? Gross. Then you go for this, right? They'll say kilojoule, 50,000 kilojoules. Okay, in aerobic glycolysis, then you should calculate how many glucose are formed. For this much kilojoules, for 50,000 kilojoules, how many glucose participated? For getting 50,000 kilojoules by oxidative phosphorylation, for getting 50,000 kilojoules by oxidative phosphorylation in aerobic glycolysis, how many glucose we required? How many glucose we required? Right? This is a question. You calculate. Right? Any doubt? Any doubt? Thank you all. Stay with us with RGR Academy. Here in RGR Academy, every Saturday, Sunday, we are conducting numerical or mathematical biology session for the NEET, for getting 360 out of 360, and also you're having difficulties in solving association reasoning, you're having difficulties in solving multiple statement question, you're having difficulty in solving graph interpretation questions. So we are engaging the various type of tough questions of the NEET Biology to secure 360 out of 360. Let's join RGR Academy to get the maximum 360 score to get AIMS, JIPMER, and other premier branded government medical college. Thank you all. Right? Take care. Bye-bye. Have a nice academic day.